In this lecture, we are going to examine the citric acid cycle. The citric acid cycle is the final common pathway for the oxidation of fuel molecules, and that means carbohydrates, fatty acids, and amino acids. It is also called tricarboxylic acid cycle, or TCA, or the Krebs cycle. It takes place in mitochondria, while glycolysis takes place in the cytoplasm. Under aerobic conditions, the pyruvate generated from glycolysis is decarboxylated to form acetyl coenzyme A, and most fuels enter the citric acid cycle as acetyl coenzyme A. The citric acid cycle itself does not generate a large amount of ATP, but it does represent the final step in cellular respiration. It removes electrons from acetyl coenzyme A, and those electrons are used to form NADH and FADH2. Most of the ATP is synthesized in the course of oxidative phosphorylation, which follows the citric acid cycle. The citric acid cycle is a cycle of oxidation-reduction reactions that results in the oxidation of acetyl group into two molecules of carbon dioxide. The process generates high-energy electrons, which are later used in the course of oxidative phosphorylation to synthesize ATP. Since it is a cycle, entry point could be any point in the cycle. The most common entry point is reaction of oxaloacetate, 4-carbon unit, with 2-carbon unit acetate in the form of acetyl coenzyme A. That gives 6-carbon tricarboxylic acid, citric acid. This tricarboxylic acid undergoes two successive decarboxylations to give first 5 and then 4-carbon unit, and that 4-carbon unit is further transformed to regenerate oxaloacetate. And then oxaloacetate, of course, undergoes another cycle. In total, one citric acid cycle generates 3 molecules of NADH, or 6 electrons for oxidative phosphorylation, 1 molecule of FADH2, or 2 electrons for oxidative phosphorylation, and 1 molecule of nucleoside triphosphate usually in the form of GTP, but it could be also ATP. In humans, about 90% of energy is produced by the citric acid cycle and subsequent oxidative phosphorylation. The citric acid cycle itself does not generate a large amount of ATP, and it does not include oxygen as a reactant. It represents the first step in cellular respiration. It removes electrons from acetyl coenzyme A and uses these electrons to generate NADH and FADH2. Each cycle generates 8 electrons, which are then utilized to reduce oxygen molecule and generate a proton gradient. And that in turn is used to synthesize ATP. So cellular respiration is shown schematically here. Fuel molecules, fatty acids, glucose, amino acids enter citric acid cycle as acetyl coenzyme A, then acetyl coenzyme A enters citric acid cycle, generates electrons, which are then used in oxidative phosphorylation to generate ATP. If we consider acetyl coenzyme A to be the entry point into the citric acid cycle, then formation of citrate from oxaloacetate is the first step of the citric acid cycle. Oxaloacetate reacts with acetyl coenzyme A. Acetyl coenzyme A, because of the thioester bond, is high-energy compound. First step is an aldol reaction to give citric coenzyme A, and then in the second hydrolysis step, citrate is formed. It's that second step that makes the overall process favorable. So product is citrate, citric acid, or under physiological conditions, citrate. The whole process is catalyzed by the enzyme citrate synthase. In the next step, citrate isomerizes into isocitrate. Enzyme is aconitase. Mechanism of isomerization is elimination of water followed by addition of water. the whole isomerization transfers hydroxyl group from carbon 3 to carbon 2 of the carbon chain. So citrate in the first step is dehydrated into cis-aconitate. That's why the enzyme is called aconitase. And then water is added in the opposite manner, so H and OH exchange places effectively, and product is isocitrate. In the third step, isocitrate is oxidized. So enzyme is isocitrate dehydrogenase, oxidizing agent is NAD. It oxidizes isocitrate on the carbon 2, it oxidizes alcohol at the carbon 2 by dehydrogenation. It removes 2 hydrogen atoms, and product is of course reduced form of NAD, NADH plus proton. The result of oxidation is oxalosuccinate, an unstable beta-keto acid which spontaneously undergoes decarboxylation. Any beta-keto acid spontaneously undergoes decarboxylation. That's hopefully something you know from organic chemistry. Product is alpha-ketoglutarate. And then in the fourth step, alpha-ketoglutarate undergoes another oxidation. Oxidizing agent is NAD and reaction with coenzyme A. The enzyme is alpha-ketoglutarate dehydrogenase complex. That complex is homologous to pyruvate dehydrogenase complex. Basically, it's identical enzyme complex to alpha-ketoglutarate dehydrogenase complex is identical to pyruvate dehydrogenase. It contains identical 3 enzymes. Reaction mechanism has the same 4 steps that were involved in reaction of pyruvate dehydrogenase that resulted in decarboxylation and formation of coenzyme A compound. The only difference is that this complex is specific for alpha-ketoglutarate, while pyruvate dehydrogenase was specific for pyruvate. So pyruvate dehydrogenase complex catalyzed decarboxylation of pyruvate and formation of coenzyme A from remaining 2 carbon unit, therefore formation of acetyl coenzyme A. Alpha-ketoglutarate complex simply is specific for alpha-ketoglutarate, which means that it's going to catalyze decarboxylation of alpha-ketoglutarate and formation of coenzyme A from the remaining now 4 carbon unit. So product is succinyl coenzyme A. In the next 5th step of the cycle, compound with high phosphoryl-transfer potential is generated from succinyl coenzyme A. So succinyl coenzyme A is high energy compound, and cleavage of thioester bond of succinyl coenzyme A is coupled to phosphorylation of GDP. So inorganic phosphate phosphorylates GDP, guanosine diphosphate. So products are succinate, coenzyme A, and a molecule of GTP. Now the enzyme is succinyl coenzyme A synthase. Some succinyl coenzyme A synthases are specific for GDP, so they phosphorylate GDP to GTP, and others are specific for ADP, and they phosphorylate ADP to ATP. Some may use either GDP or ADP. It doesn't really matter because enzyme nucleoside diphosphate kinase catalyzes exchange of gamma phosphoryl group, so it catalyzes exchange of GTP and ADP for GDP and ATP. And then in the final 6th step, oxaloacetate is regenerated by oxidation of succinate. So succinate, upon action of enzyme succinate dehydrogenase, is dehydrogenated into fumarate. In the process, FAD is reduced to FADH2. Then enzyme fumarase adds water across double bond to produce L-malate. And in the final step, NADH oxidizes malate. Enzyme is malate dehydrogenase, and products are NADH and oxaloacetate. And this oxaloacetate now can undergo another citric acid cycle. The overall process involves acetyl coenzyme A, 3 molecules of NAD, a molecule of FAD, a molecule of GDP, an inorganic phosphate, and a molecule of water. And the products are molecule of coenzyme A, 2 molecules of carbon dioxide, 3 molecules of NADH, molecule of FADH2, molecule of GTP, and 2 protons. And now we can summarize the entire citric acid cycle. So fuel molecules such as glucose, as a result of glycolysis, give pyruvate, which is then, by the action of enzyme pyruvate dehydrogenase, converted to acetyl coenzyme A. Alternatively, other fuel molecules, such as, for example, fatty acids, upon beta oxidation give acetyl coenzyme A. Acetyl coenzyme A represents entry point into the citric acid cycle. And here is the citric acid cycle itself, represented as a cycle. Acetyl coenzyme A represents the entry point, and then, if that's the case, the first step is reaction with oxaloacetate in the presence of enzyme citrate synthase, and product of the first step is citrate. In the second step, citrate, upon action of enzyme aconitase, is isomerized into isocitrate. In the following step, isocitrate, upon action of the enzyme isocitrate dehydrogenase, is oxidized and decarboxylated to the alpha-ketoglutarate, and 1 molecule of NADH is produced. That is followed by the action of alpha-ketoglutarate dehydrogenase, which decarboxylates alpha-ketoglutarate to succinyl coenzyme A, and another molecule of NADH is generated. Succinyl coenzyme A then phosphorylates molecule of GDP to GTP, which may exchange with ADP to generate ATP. Enzyme is succinyl coenzyme synthase. Coenzyme A is released, and molecule of succinate is the reaction product. Succinate in the following step is dehydrogenated into the fumarate. Byproduct is a reduced form of FAD, FADH2, and enzyme is succinate dehydrogenase. In the next step, enzyme fumarase hydrates molecule of fumarate to give malate, and in the final step of the citric acid cycle, malate, by the action of enzyme malate dehydrogenase, is oxidized to oxaloacetate. 1 molecule of NAD is reduced in the process to give NADH, and oxaloacetate now can enter another citric acid cycle. It can react with another molecule of acetyl coenzyme A and enter another citric acid cycle. This completes lecture on the citric acid cycle, and in the next lecture we are going to look at how citric acid cycle is regulated.